Welcome to Osaka. Welcome to the Osaka Metropolitan University. And I am Makoto Miyata, Emeritus from Graduate School of Science. And today I'm talking about our recent research. But sorry, I forgot to start my timer. But this is not just the scientific data, but then I made my slides a little bit more friendly to be to the all kinds of the fields. And yeah, so this is just the introduction of my research group, our students. And then I made them as the zombie, and so then revived them. And also, unfortunately, I got retired in the last March because we have the retirement year in our nation, the Japanese system. But still, I have some important projects, and also that we now I am starting up a new company, so they provide the special technology for the EM, so that we named our company. We named it as the Naniwa EM. Naniwa is the kind of traditional classical naming of the Osaka, and EM means electron microscope. And I'm now spreading, our logo, of our company, something like this one, so then two circles. So then we have in Japan a special technology where we can put the design, make the candies as we like. So then we send our logo, the design, to the candy company. So the candy company, the main, of course we pay some money, but so that they gave us the candies. So that I brought the candies. This is the you take the two pieces, and also that I am spreading my new business card. So then each piece you take one for each. Yes, you enjoy it. The candy is not so much super delicious because this is the design, and so that they don't focus on the taste, but not bad. So then when you feel very hungry, you can survive. And this is what is right. What is right? I'm saying that life of you was not that one. The life was dead. So then what is right? So then I summarize understanding of the biology. So here you can see the materials, chemicals related to the life. And when understanding in the cell, the materials packed, and some reactions happen, occur. So then they input, update, this material and the energy and the information from the environment. So then they do some complicated reactions. So then they make somehow related, somehow the similar thing. So that this is called the reproduction. And also that they, live cells, they give some action to the environment. So then some biologists are challenged to reproduce something right. This is a kind of typical popular, challenge form. So that those kind of biologists that try to make some content, something like life inside the cell membrane. So then somehow they want to start real life, something like that, but nobody achieves it. So then you're understanding that this is the challenge, too much challenge, I'm saying. So the too much work, so the too much difficult. So my suggestion is we should lower the hurdle. This is our our idea. Then here is the existing light. So then we can remove just activity. So now we have the dead body, but still they keep the old content of the light, but somehow they are dead. So then somehow we revive the dead cell body. So then we can say that what is the boundary between the material and the light. For example, if this is something living thing, so then we make it, if this is something dead thing, so then we revive it, so then kill it, revive it, kill it, so then we can understand the boundary. That is our idea. Interestingly, here the previous some biologists did something like that. One famous and very exciting thing was done by Robert Cornish at the UC Berkeley in 1900s. So then he tried to revive dead human body and dead dog bodies. So then he was somehow achieved, but we don't know that in the modern science. But people believed it, and he became a superhero. And it was picked by the documentary film like that. You can access to that through the YouTube even now. Because that is because the world was going to the starting the World War I. So then people were worried. Many people will die soon. Anyway. So then, I forgot to put the. So then you can see the many YouTube videos. So then we can try it now. So then just I found yesterday the kind of typical thing. I show you one second. Not this one, but most of the QR codes are printed on my business cards, so you can enjoy it. I cut connection. I stopped connection. My fault. So again, I have some margin for time.
What if we could?
This is just short one, but you can find many of such kind of things. This is just imagination of the robot bodies. Yeah, there are the differences. So this is kind of the seesaw, the facility to revive the dead bodies. If the principle is too much simple, so if they move the blood flow, of the dead body, so then somehow they become reactivated. But actually, it's a kind of real strength if we think from the current modern biology. But somehow, he was really talented person, the doctor. So that he developed many important techniques to save the lives. Then you can save many something like that. Anyway, then you can use the micro. Yes, and another very recent sample is this was recent sample is done these members, and they revived the pig chopped head. And it was published in that Nature magazine, and also it got autometrics, this high score. So then I care about autometrics because it's the evaluation or the impact on the impact of the science on the natural human drive. So it's based evaluation is based on the X post anyway. But human dog pigs are too much, and it's sometimes problematic. So then we thought about minimal life. Then what minimal life is? And we focus on the minimal synthetic bacterium JCVI-C3. So then the JCVI John Craig Venter Institute was governed by the Professor John Craig Venter. It's very direct. Anyway, so then John Craig Venter is the fame of the genome, decoding of human. And he focused on the, after he determined the human genome sequences, so then he focused on to make the minimal cell, minimal living thing, just near the border of the material. So then they achieved it and published it in Science magazine in 2016. And I was so much impressed about this paper because they removed the DNA from the first growing bacterium and then determine which materials are essential set for life. So then this is the image was taken by our company, really special technique. The red one, the red box one is we call this the minimum synthetic material as SYN3 for short. So the SYN3 has only 438 proteins and 39 RNAs. Please you remember the biology classes in the high school. And if the cell is very small, even compared to the acetyl chloride, standard strain, and also the protein numbers. The number of the proteins are 10 times smaller than the usual acetyl chloride. So then we say that this paper is landmark, and so that they define what is the minimal material set for life. But unfortunately, Craig Venter passed away in this year. He didn't get the Nobel Prize, but that's okay. So then they define the minimal set of the material, but what else? So then we read many previous papers. So then we suggest 3 physical factors are required other than materials. So then here is a schematic. So then anyway, so then here is a schematic, so that you can see the membrane, so that it contains the DNA and also the other materials like that, the ATP, the membrane potential, RNA. So then I'm saying the 3 other factors needed for life. So the first one is high density coding organization of the materials. And I put I present it by the broken lines. And the second one is that still that they have some diffusion by energy. So it's very unusual. And also the directing from DNA to keep the condition of the A and B. So then now I'm showing you our recent data showing to make the zombie of the SYN3. So this is a return, divide them to life. And this is the design of our experiment. So regarding the DNA activity. So then this is the SYN3 schematic. So then we remove the DNA by expressing the hydrolyzing enzyme. So then we can get the zombie, but this may be have the activity to revive to life. So then we put back the different types of different DNA into the live zombie. So then we may get the revival. And also that if this is a live zombie, so there should be the dead zombie. So then we are very interested in the boundary of the life, right, and materials. This is the kind of material anyway. So then I'm discussing about this process. So then we introduce the hydrolyzing enzyme at the time zero, and this is the cell appearance. And you can see the cell appearance doesn't change so much, but if we visualize DNA, the DNA disappears. So it means the cell, the sensory is zombified. And we can see we can show the number of the live sensory. So then we counted the number, the live cell number, and here is the start of zombification. So then you can see that immediately after the induction of the hydrolyzing enzymes, the number of the live sensory decreased immediately. So then I'll show you the revival process. It's a little bit difficult to understand. So then if you write, I'll explain anytime. Anyway, we focus on these 4 points, and then we recover the cells and put back DNA, but it has the new, character. So then we can say that this, the sensory is original sensory, and this sensory is the divided sensory. So then we measure the x-axis is the zombification time, and y-axis the number of the revivals. So then this is just at the time zero. Sorry, yes. I tried this one. No, in the anyway. So then. So that time zero. So then this number of the cell, the C3 cells, it's the revived as the new type of the C3. But here they are zombificated. So then even the zombificated C3, so some these numbers of the cell became new C3. It means revival from the live zombie. And also and the number decreased with the time of the zombification. So then we can say that this kind of schematic, the sensory was zombified. The zombie is now that here the zombie was more live, but even if just the live zombie with the DNA, so that they gradually became to the dead zombie. So then, and also this is kind of too much biology, but we measure the condition of the energy of the cell with the DNA, and our conclusion is that somehow energy state decreased with time, so that they became unable to revive. Anyway, so then we can appear that we visualize the 2-3 boundaries. The first one from the sensory, the live to the somehow dead, but the somehow dead to live, and the somehow dead to the real dead. Yes, so then somehow that this is we can appear that we achieve the first small short goal of our interest. So then, and also that this is just as I told you that I get retired. So many people that came to our university. So then we did the kind of the big party of and including my final lecture. So then I appreciate all of the collaborators and and my students. So that I share a video through the the YouTube, the link is printed on my business card. Actually, I came with some the Michael Jackson, so the Michael Jackson thriller. But the people, we worried that if we put the Michael Jackson the music, so they will remove from YouTube. So they will replace the sound. So then, of course, I appreciate all of your attention very much. So that any kind of question is very welcome. Thank you. Go ahead.
So, this is kind of a weirdly formatted question, but my personal thoughts are that, you could bring a single cell back to life pretty well because it only has one, it's only one cell. It doesn't have to collaborate with anything else. But once you start going to a multicellular organism, like a dog or a person, it's very hard to bring it back to life because each cell individually is dying and the chemicals are different. Like, what is your thoughts on that?
Right. So it's very interesting thing, and also that it's easy to answer. The kind of a long discussion. Mm-hmm. So for example, so then if I become really the scientist, biologist. So then we focus on the boundary, live cell and live dead cell, material and cell. So we focus on just the single molecule, the single cell thing. But of course, I agree with you. So then death of the single cell is different from the death of the multicellular things. That's the big question. So that's just I'm saying that it's so many interesting things. For example, when we die, so that we can't respond anymore, so that we don't need anything. For example, but there are kind of very interesting argument. So that if you we keep the dead body in the box, so under the ground. So then, for example, so that in sorry, I'm not very familiar for that. It's a kind of Dracula something. Yeah. So then they, take back the dead body. Sometimes, the nail grew and the hair grew. So it means, so it's kind of that the old people, they were so much, scared by that, but actually it's a kind of a usual thing. So if we are totally dead, so then the brain is not working at all, but the smaller cells, many cells are still they are alive. So it's a kind of the big argument. So then we are, the big animal is very easy because they have some the consciousness. So then we can say that he loses his consciousness. We can say that he is dead or something. But if you think about it, for example, even they don't have the clear consciousness, some multicellular things, for example, so the multicellular, the slime balls, so there's something, so there's some parts that died, but still some other parts that are alive. So it's multicellular, things, it's the boundary is more complicated. I agree. But I'm curious, I'm interested in, cell itself. But this can be applied for the, medical science, because if we can keep the dead body, so somehow we recover the tissue, we can get the better surgery. Currently, the surgery should be done, just always the cells are actively moving, working. So it's completely strange if we think about, if we fix our computer, so we should turn off the computer and replace the parts. So we can fix the computer. So that's the same thing for the living things. Then we believe that our technology, our research will give some impact on the medical science. Yes. Sorry, I can't answer your question directly.
No, that's okay.
Yes. Go ahead.
This is a little bit more of a philosophical question, but you've been doing a lot of research on death. How has that affected your own views on your morality or mortality and your potential death? Has that changed it at all?
Sorry, I don't understand. Really, it's a little bit,
Has researching death
changed your views on death?
Yeah. It's not also the direct answer, but the formally we were working on I've been working on the origin of the motility. So then we succeeded to put the small genes into the SIN3. So then we got we gave enabled they to move. So then at the meantime, so that somehow I got more interest. At that time we were working the boundary between non-motile and motile. So then somehow I keep in mind that someday I will do the boundary data line. So then, but interestingly, 2022, I mostly died by stroke. So then, after that, somehow I survived. So then, after I came back from the hospital, somehow I prefer to the switch of research from motility to the life and material. So I can't answer to your question, but somehow related to that. Yes, other questions, please go ahead.
You talked about your short-term goals, but what are the long-term goals? What are you guys looking forward to next?
Sorry, that you were asking about the origin of life. No.
No. You said you talked about your short-term goals and how you met them. What's next? What are you guys looking forward to?
Yeah. No, yes. So then I just showed the boundary between the. Now that we are writing this paper because this this data really big the discovery. So then we are writing this one. So they usually if we publish it in the Nature magazine, the Science magazine, or somehow similar impacts, then we need to do a lot of the experiments. And also that we are now doing I showed you just the result of the DNA direct thing, but so that we are also working on the fluidity of the high concentration the cells, and yes, and so that actually the people people believe that the materials are so much packed in the living cell. But actually, it was based on the really, all the days, data. So that we are, doing that kind of research with, current technology. And so then the answer is we are so many things to do even the experiment. So then we are doing we are still working the other, two physical factors, and also now my focus is that I didn't say that show the detail, but we want to continue sustainably. So then we want to provide this our technology, and at the same time, so then as I told you, so that people, biologists are trying to reconstruct from the kind of nothing to get suddenly the living thing. But if we kill the existing cells, so then we revive them, so that it can be much more easy. So then I want to establish that kind of field. So then by using that, of course, I want to contribute the understanding of the the material right boundary, but also that I want to establish those kind of the fields in the field of the biology and the biophysics. That's our focus, our foresight now. Yes. Is there any questions? 私が司会していいんですか？終わりとか言ってるんですか？Ah, please go ahead.
Not question, but it seemed like he's done doing presentation.